Welcome to Excellent Grades Academy. This is Dr. Byson AM. This video brings to you physiology multiple choice questions under muscle and nerve physiology. So let's get into these questions. Okay, question 1 says, which of the following characteristics is shared by simple and facilitated diffusion of glucose? A says, occurs down an electrochemical gradient. So this is correct. Okay, so simple and facilitated diffusion both occur down an electrochemical gradient. Number 2 says, during the upstroke of an action potential, A, there is net onward current and the cell interior becomes more negative. So this is false because the cell's interior becomes more positive. B says there is net outward current and the cell interior becomes less negative. So we know that the net current moves inside. So there is net inflow or inward current. Number 6 says there is net inward current and the cell interior becomes more negative. The cell interior becomes more positive. This says there is net inward current, yes, and the interior becomes less negative. So the answer here is D. Let's move on. Number 3, solutions A and B are separated by a semi-permeable membrane that is permeable to potassium but not to chloride. Solution A is 100 millimolar calcium chloride and solution B is 1 millimolar potassium chloride rather. Which of the following statements about solution A and solution B is true? So potassium ions, A says potassium ions will diffuse from solution A to solution B until potassium of both chloride is 50.5 millimolar. No. B says potassium ions will diffuse from solution B to solution A until the potassium of both solutions is 50.5 millimolar. No. C says potassium chloride would diffuse from solution A to solution B until the potassium of both solutions is 50.5 millimolar. No. This says potassium will diffuse from solution A to solution B until a membrane potential develops with solution A negative with respect to solution B. So the answer here is D. This is the answer. 4. The correct temporal sequence for events at the neuromuscular junction is A. Action potential in the nerve cell, depolarization of the muscle end plate, uptake of calcium into the presynaptic nerve terminal. No. B. Uptake of calcium into the presynaptic terminal. Yes. Release of acetylcholine. Yes. Then depolarization of the muscle end plate. Correct. So the answer here is B. So number 4 is B. Number 5. Which characteristic or component is shared by skeletal muscle and smooth muscle? A. Thick and thin filaments arranged in sarcomeres. So no, we know that smooth muscles are not arranged in sarcomeres. B. Troponin. That's false because we know that in smooth muscle there is no troponin. C. Elevation of intracellular calcium for excitation contraction coupling. So this is correct. We know that in both muscles there is elevation of calcium for the excitation contraction coupling. This is spontaneous depolarization of the membrane potential. This is false because this spontaneous depolarization only occurs in smooth muscle and then it doesn't occur in skeletal muscle. High degree of electrical coupling, B. No. So 6. Repeated stimulation of a skeletal muscle fiber causes tetanic contraction because the intracellular concentration of which solute increases and remains at high levels. The intracellular concentration of which solute increases and remains in high levels. Okay, so the answer here is E. So calcium is the one that increases and remains at high levels. So the answer is E there. Number 7. Solutions A and B are separated by a membrane that is permeable to calcium and impermeable to chloride. Solution A contains 10 millimolar calcium chloride and solution B contains 1 millimolar calcium chloride. Assuming that 2.3 RT over F is 60 millivolts, calcium will be at electrochemical equilibrium when A solution A is plus 60 millivolts, B solution A is plus 30 millivolts, C solution A is negative 60 millivolts, D solution A is at negative 30 millivolts. So the answer here is this one. All right, so you need to know how to calculate the potentials there for solutions. 8. A person with myasthenia gravis, this is a very common condition in physiology, you need to study about this, notes increased muscle strength when he is treated with an acetylcholinesterase inhibitor. The basis for this improvement is increased A, amount of acetylcholine released. The basis for this improvement is increased A, amount of acetylcholine increased from motor nerves. B, levels of acetylcholine at the muscle end plates. Yes, so there is increased levels of acetylcholine at the muscle end plates. That's why there is increased muscle strength in myasthenia gravis. Number 9. In error, a patient is infused with large volumes of a solution that causes lysis of his red blood cells. The solution was most likely, so we know that the solution was most likely an hypotonic urea solution. Why? Because we know hypotonic solutions are the ones that go into cells. 10. During a nerve action potential, a stimulus is delivered as indicated by the arrow shown in the following figure. This is the arrow here. Okay, this is the arrow. In response to the stimulus, a second action potential, so A, of smaller magnitude will occur, no. Of normal magnitude will occur, no. Of normal magnitude will occur but will be delayed, no. E will occur but will not have an overshoot. E, so the answer here is E. So here, a second action potential will not occur. The second action potential is not going to occur. Why? Because this nerve is going to enter refractory period. So during the refractory period, no action potential is going to take place. Number 11. Solutions A and B are separated by a membrane that is impermeable to urea. Solution A is 10 millimolar urea and solution B is 5 millimolar urea. If the concentration of urea in solution A is doubled, the flux of urea across the membrane will increase, so it will by 3 times, so it will triple. The flux of urea across the membrane will triple. 12. A muscle cell has an intracellular sodium of 14 millimolar and an extracellular sodium of 140 millimolar. Assuming that 2.3 RT over F is 60 millivolts, what would be the membrane potential B if the muscle cell was permeable only to sodium? So the answer here is D. Okay. It would be plus 60 millivolts. Okay, the reason why it's plus 60 millivolts is because sodium is positively charged. Number 13. So this diagram here, the diagram of a nerve action potential, applies to question 13 and question 15. So study this. Okay, so this is an action potential curve of a nerve cell. So nerve cell action potential. We know that in a muscle cell, the action potential is different. Okay, so now questions follows at which labeled point on the action potential is the sodium closest to the electrochemical equilibrium? So the answer, let's refer to the diagram. It's at 5. So at here, that's when the sodium is closer to the, so the answer here is E. At this point, the sodium is closest to the electrochemical equilibrium. 14. What process is responsible for the change in membrane potential that occurs between 0.1 and 0.3? Okay, so 0.1 and 0.3. So 0.1 and 0.3. So here we know that there is depolarization and there is movement of sodium in the cell. So the answer here is A. So there is movement of sodium into the cell. Number 15. Which process is responsible for the change in membrane potential that occurs between 0.3 and 0.4? So let's see 0.3 and 0.4. So 0.3 and 0.4, 0.3 and 0.4. So 0.3 and 0.4, we know here that sodium is moving outside of the cell. Okay, so the answer is D. There is movement of sodium outside of the cell. All right, let's go to 16. The rate of conduction of action potentials along a nerve will be increased by A. Stimulating the sodium potassium ATPase pump? No. Inhibiting the sodium potassium pump? No. Decreasing the diameter of the nerve? No. Myelinating the nerve? So yes, we know that when you myelinate the nerve, the rate of action potential speed is going to increase. Okay. Number 17. Solutions A and B are separated by a semipermeable membrane. Solution A contains 1 millimolar sucrose and 1 millimolar urea. Solution B contains 1 millimolar sucrose. The reflection coefficient for sucrose is 1 and the reflection coefficient for urea is 0. Which of the following statements about these solutions is correct? Okay, so the solutions A has a higher effective osmotic pressure than solution B. No. Solution A has a higher effective osmotic pressure than B. No. Solution C. Solutions A and B are isosmatic. No. Then D. Solution A is hyperosmotic with respect to solution B. And the solutions are isosmotic. So the answer here is D. 18. Transport of D and L-glucose proceeds at the same rate down an electrochemical gradient by which of the following processes? Simple diffusion. So this is the answer here. So the answer is simple diffusion. Number 19. The permeability of a solute in a lipid bilayer will be increased by an increase in the molecular radius of the solution, of the solute. No. Oil or water, partition, coefficient of the solution. So the answer here is B. 20. A drug completely blocks sodium channels in nerves. Which of the following effects on the action potential would it be expected to produce? A. Block the occurrence of action potentials. Yes, it will block the occurrence of action potentials because we know that for an action potential to occur, sodium has to enter the cell. 21. At the muscle end plate, acetylcholine causes the opening of 21. So A. Sodium channels and depolarize towards the sodium equilibrium potential. No. Potassium channels and depolarize towards the potassium equilibrium potential. No. Calcium channels and depolarize towards the calcium. No. Sodium and potassium channels and depolarize to a value halfway between the sodium and potassium equilibrium potential. So the answer here is D. 22. An inhibitory postsynaptic potential depolarizes the postsynaptic membrane by opening sodium channels. No. Depolarizes the postsynaptic membrane by opening potassium channels. No. Hyperpolarizes the postsynaptic membrane by opening the calcium channels. No. Hyperpolarizes the postsynaptic membrane by opening the chloride potentials. So the answer here is D. 23. Which of the following would occur as a result of the inhibition of the sodium-potassium ATPase? So A, decreased intracellular sodium concentration. No. B, increased sodium-calcium exchange. No. Increased intracellular calcium concentration. So the answer here is C here. So increased intracellular calcium concentration would occur as a result of inhibition of the sodium-potassium ATPase. All right, 24. Which of the following temporal sequences is correct for excitation contraction coupling in skeletal muscles? A, increased intracellular calcium action potentials in the skeletal muscles, in the muscle membrane, cross bridge formation. So no. Action potential in the muscle, depolarization of the T tubules, and release of calcium from the sarcoplasmic reticulum. So this is what is going to occur. 25. Which of the following transport processes is involved if transport of glucose from the intestinal lumen into the small intestine cell is inhibited by abolishing the usual sodium gradient across the cell membrane? A, simple diffusion. B, facilitated diffusion. C, primary active transport. So the answer here is co-transport, because we know that sodium will carry glucose in secondary active transport, which is co-transport. 26. Which of the following events occurs before depolarization of the T tubules in skeletal muscles in the cell mechanism of excitation contraction coupling? A, depolarization of the sarcolemma membrane. So this is correct. So there is depolarization of the sarcolemma membrane. 27. Which of the following is an inhibitory neurotransmitter in the central nervous system? So the answer here is gamma aminobutyric acid. 28. Adenosine triphosphate ATP is used indirectly for which of the following processes? A, accumulation of calcium by the sarcoplasmic reticulum. No. Transport of sodium from intracellular to extracellular. No. Fluid. No. Transport of 1 potassium from extracellular to intracellular. No. So a transport of hydrogen ions from parietal cells to the lumen of the stomach. E, absorption of glucose by intestinal epithelia. So the answer here is this, E. 29. Which of the following causes rigor mortis? So rigor mortis: A, no action potentials in motor neurons. No. An increase in intracellular calcium level. No. An increase in intracellular calcium level. A decrease in intracellular calcium level. No. An increase in adenosine triphosphate level. No. So rigor mortis is caused by a decrease in ATP levels. Rigor mortis simply means that there is rigidity in the body and there is no movement. 30. Degeneration of dopaminergic neurons has been implicated in A, schizophrenia. B, Parkinson's disease. So the answer is Parkinson's disease. So in Parkinson's disease, you have degeneration of the dopamine neurons, meaning that there is no inhibition of neurons. So you have involuntary uncontrolled movement because there is uncontrolled firing of nerves in Parkinson's disease. 31. Assuming complete dissociation of all solutes, which of the following solutions would be hyperosmotic to 1 millimolar of sodium chloride? 1 millimolar of glucose? No. 1.5 millimolar of glucose? No. C, 1 millimolar calcium chloride. So this is the answer here. 32. Secretion of hydrogen ions by gastric parietal cells occurs by which of the following processes? So it is by primary active transport. You need to know this. Primary active transport. Okay, so that was the physiology marathon that included nerve physiology, muscle physiology, and transport across the cell membrane. Revise, know these questions, know this data. You can register for lessons on the number that is projecting on your screen. I'll see you in the next video.